A shark can lose a tooth in violence and not care, not because it heals better, because the replacement was already waiting. Now compare that to you. Lose one adult molar and your body does something strangely final. It bleeds, it seals, the gum closes, the bone remodels, the bite adjusts around the absence. No new molar rises from underneath. No third dentition starts. No hidden reserve tooth rotates into place. The wound heals, but the tooth is gone. That is the real mystery, not why sharks make so many teeth. Why do humans stop? Hey folks, I'm Dr. William Gray, aka Wiggle Will. I'm an oral and maxillofacial surgeon with an MD, a DMD, and an MBA. And this is Body Logic, where we explain the strange, painful, powerful, and misunderstood parts of biology without turning science into either a lecture or a conspiracy. Quick heads up before we start. Some episodes use my real voice, and some use an AI voice trained on mine. Either way, the science and the judgment are always mine. Let's get into it. The unsettling part is that the human jaw is not completely ignorant of how teeth are made. It once knew how. It did it before you were born. It did it when your baby teeth formed. It did it again when your permanent teeth formed. Even after that, fragments of the old program can remain. But in humans, that program is no longer arranged as a living replacement system. It is broken apart, muted, controlled. When pieces of it wake up in the wrong way, they do not produce a clean new tooth. They can produce cysts, tumors, or disorganized odontogenic tissue. This is not a story about sharks being better than us. It is a story about two different solutions to damage. One animal keeps replacing the blade. The other protects the bite. Somewhere between those two strategies is one of the biggest questions in regenerative dentistry.
3 myths to retire. First, sharks have unlimited teeth. No. Second, sharks do not get cavities because shark enamel is magical. No. The bigger reason is time. Third, humans cannot regrow teeth because the program vanished. Not exactly. The machinery is partly present. If you understand the break, you understand both the promise and the danger.
Why can a shark keep making teeth while a human jaw learns to live with absence? To answer that, we have to start before teeth look like teeth, before enamel, before roots, before eruption. A tooth begins as a conversation between tissues. The surface tissue folds inward. The tissue beneath answers. A signal becomes a place becomes a bud becomes a shape. Eventually, that shape becomes a tooth. The structure that begins this process is called the dental lamina. It is not a tooth. It is the developmental line from which teeth are initiated. Sharks use it. Humans use it. Many jawed vertebrates use some version of this same ancient developmental logic. The signals involved are not random. WNT helps initiate tooth formation. Beta-catenin helps transmit that signal. BMP signals help shape the tissue. Sonic hedgehog helps organize growth and position. FGF signals help keep communication going between epithelium and mesenchyme. This is not one magic molecule. It is a controlled sequence. The right signal, in the right place, at the right time, held back in the surrounding tissue so the pattern does not dissolve into chaos. A tooth is not just something that grows. It has. So the real question is not why sharks have a tooth program and humans do not. The sharper question is why sharks keep the program open while humans close it. Here is the shark difference. On the tongue side of the jaw, the successional lamina persists as a living epithelial stem cell niche. This niche contains progenitor-like cells and proliferative signaling. Instead of making one replacement tooth and disappearing, the tissue keeps cycling. Think of it as a start-stop engine. WNT activation begins a new bud. But if activation spread everywhere, the system would not make teeth in orderly rows. It would make noise. Sonic hedgehog helps organize boundaries and spacing. BMP signals help shape the structure. FGFs keep it going. The point is the right signal, in the right place at the right time. Initiate, confine, shape, move, initiate again. This is biology with rhythm. Shark teeth are not socketed into bone like human teeth. Humans have thecodont attachment in sockets with periodontal ligament. Sharks have cartilaginous jaws and connective tissue. This is why a shark jaw looks like a conveyor belt. Teeth form behind the functional row, rotate outward, mineralize, and move forward. The visible tooth is only the front edge of the system. Behind it are younger teeth and buds. The mouth is not just using teeth, it is manufacturing them. This is why a shark jaw looks like a conveyor belt. That makes replacement easier. A disposable tooth belt works when each tooth does not need a long-term bony socket, periodontal ligament, and precise relationship. A great white may cycle through thousands to tens of thousands of teeth. The count depends on species and rate. The numbers are dramatic, but they are not infinite. Replacement rate varies by species, temperature, and diet. Some turn teeth over quickly. Others take weeks. Some sharks are even stranger. Cookie cutter and kitefin sharks can shed whole lower tooth plates together, likely recycling minerals after swallowing them. So myth 1 falls apart. Sharks do not have unlimited teeth. The critical word is controlled. If WNT stayed high everywhere, the lamina would create disorder. Shark replacement works because activation is local and temporary. The technical language sounds complex, but the concept is simple. One tissue says, build tooth here. The tissue beneath answers, shape it this way. If one is gone, biology mostly shrugs. Same planet, same vertebrate history, completely different dental outcome. Why? The conveyor belt metaphor is useful, but incomplete. The belt is manufacturing the parts as they travel. Early buds are soft, later teeth mineralize. Human teeth are engineered for long service inside sockets. Shark teeth are cutting edges. Brilliant for a predator, but terrible for a human molar that needs stability for decades. The surprising part is that these rests are not always blank. They can express markers associated with tooth making. The workers remain, but the floor is gone. This is where the story becomes clinical. Dormant remnants can misbehave. Oral surgeons know this truth well. The shark tooth factory never really closes. In humans, it is locked. Learning to unlock it safely is the real miracle. Controlled teeth. Shark replacement is not just a feature. It is a whole mechanical and developmental system. Everything fits together. When epithelial remnants reactivate the wrong way, they can contribute to cysts and tumors, including ameloblastoma. These are tissue losing its normal restraint. Myth 2 also needs correction. Sharks are not protected because their enamel is magical. The practical answer is time. A tooth shed soon has less time for decay. And in humans, the break is usually a good thing. Without it, quiet rest could become pathology instead of regeneration. Stability over endless novelty. Apoptosis matters because shutdown is active. The lamina is dismantled. Cells receive death signals. The islands lose the geometry that once told them where a tooth belongs. The human story is controlled suppression. Mammalian chewing depends on precise occlusion. Continuous replacement would constantly disrupt that geometry. Imagine replacing one gear in a watch every week and still expecting it to keep perfect time. That is the mammalian trade-off. But regenerative dentistry is asking if we can borrow part of the shark logic without the shark problem. It belongs to the same family of questions. Anti-USAG1 research is interesting because it points toward a developmental break. The idea is to release a break carefully in selected patients. Making a tooth requires a controlled developmental sequence. Architecture must be maintained. 3 myths retire here. Sharks do not have unlimited teeth. Humans did not lose the program, they suppressed.
A tooth is not just enamel on top of bone, it is a negotiated organ. Epithelium and mesenchyme pass instructions back and forth until crown shape, root pattern, and mineralization all line up. Here is the shark difference, the successional lamina. On the tongue side of the jaw, the dental lamina persists as a living epithelial stem cell niche. It contains progenitor cells marked by SOX2, BMI1, beta-catenin, and other proliferation signals. Instead of making one replacement tooth and disappearing, this tissue keeps cycling. Think of it as a start-stop engine. Wnt activation begins a new tooth bud, then Sonic Hedgehog helps repress Wnt in the plane of the tissue, so the next bud forms in the correct position. This is the spatial discipline. If Wnt simply stayed high everywhere, the lamina would not make a neat replacement row. It would make chaos. The shark system works because activation is local and temporary. A bud starts, neighboring tissue is held back, and the next generation waits its turn. This is why the conveyor metaphor is useful but incomplete. The belt does not merely carry finished parts. It is manufacturing the parts as they travel. Early buds are soft epithelial and mesenchymal structures. Later teeth mineralize, then the functional row takes the bite forces, fails, sheds, and gets replaced from behind. The attachment system makes that possible. Human teeth are engineered for long service inside sockets. Shark teeth are cutting edges on a replaceable membrane. That is brilliant for a predator that breaks teeth on prey. It would be terrible for a human molar trying to maintain a precise bite for 40 years. The biology is therefore not just more versus fewer. It is a whole mechanical system. Stem niche, signaling rhythm, connective tissue attachment, jaw cartilage, diet, and replacement timing all working together. Now look at the human side. During development, humans absolutely use a dental lamina. We form deciduous teeth, then permanent successors, and the molars form sequentially. But after that, the lamina does not remain as an active conveyor. It fragments. Cells undergo programmed apoptosis. The once continuous tooth-making band breaks into scattered epithelial islands in the gingiva. These are called dental lamina rests. In plain English, the workers are still standing around in the rubble. The factory floor was dynamited, but not every worker left the building. That is why the story is not only evolutionary, it is clinical. When these epithelial remnants reactivate the wrong way, they can contribute to odontogenic cysts and tumors, including ameloblastoma. But biologically, they reveal the same uncomfortable truth. So myth 2 also needs correction. Sharks are not cavity-proof because their enamel is magic. The practical answer is turnover. A tooth that is going to be shed soon has less time to become a long-term decay problem. Humans chose the opposite strategy. Keep the teeth, fit them precisely, maintain them for decades. The machinery is not gone. The brake is on. And in humans, that brake is normally a good thing. Without it, the quiet rests of the dental lamina can become pathology. The word apoptosis matters because shutdown is active. The lamina is not simply forgotten, it is dismantled. Cells receive death signals, the sheet loses continuity. That loss of architecture is as important as loss of signaling. A single surviving epithelial island is not automatically a tooth factory. A real tooth needs position, polarity, mesenchymal instruction, mineralizing cells, eruption path, and integration with the bite. That is the safety problem. The same residual developmental potential that makes regeneration imaginable also explains why blind reactivation would be dangerous. If you wake the wrong cells in the wrong place with the wrong signals, you do not get a useful third molar. You may get a cyst, a tumor, or disorganized odontogenic tissue. So the human story is not failure, it is controlled suppression. The jaw chooses stability over endless novelty. Why would mammals accept that trade? Occlusion. Mammalian chewing depends on precise cusp and fossa contact. Upper and lower teeth interlock. Molars crush, grind, shear, and guide the jaw through repeatable paths. Continuous replacement would constantly disrupt that geometry. Imagine replacing one gear in a watch every week while still expecting the watch to keep perfect time. Nursing also changes the developmental schedule. The payoff becomes 2 highly tuned sets, not a lifetime of disposable blades. Humans are diphyodont, 2 dentitions. Sharks are polyphyodont, many replacement cycles. That is not a defect. It is an evolutionary bargain. But regenerative dentistry is asking whether we can borrow part of the shark logic without importing the shark problem. The cat-shark work maps a Wnt, sonic hedgehog, BMP, and FGF circuit that might be coaxed in human remnants. And that is why the anti-USAG1 work is so interesting. Paragam Biopharma in Japan moved an antibody approach into a phase 1 trial beginning in 2024, aimed at congenital tooth agenesis. The concept is not make humans into sharks. The concept is release a developmental brake carefully enough to allow a new tooth program to start. This is early. It is not a consumer tooth regrowth product. The future version, if it works, will probably not look like pressing a button and instantly growing a perfect adult tooth. It will look more like developmental steering. That is exactly why sharks matter. They show the natural version of a program that stays organized across thousands of cycles. There is one more layer. If the shark story sounds simple, the human story is the warning label. The most important thing to understand is that regeneration is not one switch. A tooth is an organ. It needs a surface epithelium, a responsive mesenchyme, a growth field, and integration with the bite. In a shark, the successional lamina is still a line. It has direction. It has polarity. That geometry tells each new tooth where it belongs. In an adult human jaw, the remnants are islands. Islands are not conveyor belts. This is the real reason tooth regeneration is hard. You are not only asking cells to divide, you are asking them to organize. Where does a new bud start? How does the crown know what to become? How does it move without wrecking the bite? The goal will be one correct tooth in one correct place, grown slowly enough and safely enough that the jaw accepts it as its own. That would be the real miracle, not endless teeth, controlled teeth. Evolution did not delete the tooth factory, it silenced it. Next episode, why elephants and manatees solve the tooth problem in a completely different way. Real biology, real mechanism.